This is Biochemistry, Part 3. We start with the laws of thermodynamics. There are 4 of them that you need to know. The Zeroth Law of thermodynamics. If 2 systems are in thermal equilibrium with a third system, they are in thermal equilibrium with each other. This law helps define the concept of temperature. The first law of thermodynamics. When energy passes as work, as heat, or with matter into or out from a system, the system's internal energy changes in accord with the law of conservation of energy. Equivalently, perpetual motion machines of the first kind, machines that produce work with no energy input, are impossible. The second law of thermodynamics. In a natural thermodynamic process, the sum of the entropies of the interacting thermodynamic systems increases. Perpetual motion machines of the second kind, machines that spontaneously convert thermal energy into mechanical work, are impossible. And then we also have the third law of thermodynamics. The entropy of a system approaches a constant value as the temperature approaches absolute zero. With the exception of non-crystalline solids, the entropy of a system at absolute zero is typically close to zero and is equal to the natural logarithm of the product of the quantum ground states. A cofactor is a non-protein chemical compound or metallic ion that is required for a protein's biological activity to happen. These proteins are commonly enzymes, and cofactors can be considered helper molecules that assist in biochemical transformations. Cofactors can be subclassified as either inorganic ions or complex organic molecules called coenzymes. Coenzymes are mostly derived from vitamins and other organic essential nutrients in small amounts. In an exergonic reaction, the potential energy of the products is less than that of the reactants. Exergonic reactions are spontaneous. The example of exergonic reactions that occur in our body is cellular respiration. This reaction releases energy which is used for cell activities. Endergonic reactions require energy and therefore are not spontaneous. Photosynthesis is usually the classic example of an endergonic reaction. In an endergonic reaction, the change in G is greater than zero, which is positive, while in an exergonic reaction, the change is less than zero, and so it's negative. The active site refers to the specific region of where a substrate binds and catalysis takes place or where chemical reaction occurs. It is a structural element of protein that determines whether the protein is functional when undergoing a reaction. This structural element will be accordingly shaped to the structure of the enzyme at work on it. The active site is made of residues that can form bonds with the substrate, particularly at the binding site. Often, an enzyme would have only one active site that will fit with only one specific substrate. At the binding site, the residues form temporary bonds with the substrate to form a complex. In competitive inhibition, there are 2 different substrates that compete for an active site on one enzyme. Both substrates cannot bond to the enzyme. The substrate in greater concentration will most likely bond. A non-competitive inhibitor is a substance that interacts with the enzyme, but usually not at the active site. The effect of a non-competitive inhibitor is to change the shape of the enzyme and thus the active site so that the substrate can no longer interact with the enzyme to give a reaction.